Hello, my name is Bruna Burssed. I'd like to thank the organizers for inviting me to present at the symposium. This is my presentation entitled Complex Constitutional Rearrangements Characterized by the Combination of Optical Genome Mapping and Long-Read Sequencing. Structural variants are alterations that result from breaks and rearrangements in the chromosomes, and these include translocations, deletions, duplications, and inversions. Among the structural variants, we can also have complex rearrangements, which we have two kinds. The first one are the complex genomic rearrangements, which include structural variants with more than one junction or more than one structural variant in cis. The other type of complex rearrangement are the complex chromosomal rearrangement, which involves structural variants with more than 3 breakpoints that involve more than 2 chromosomes. These complex rearrangements are believed to be formed in a single catastrophic event that leads to multiple structural rearrangements. We have 3 mechanisms that fit this description. The first one is known as chromotripsis, where we have a single chromosome that shatters and then is reassembled through non-homologous end joining, forming an entirely new chromosome that can present deletions. The second mechanism is known as chromoanasynthesis, and just like the first one, also involves a single chromosome that undergoes recombination through a replication mechanism, such as phosphatase, and then forms, again, an entirely new chromosome that can present duplications, triplications, deletions, and inversions. The final mechanism, unlike the other two, is known as chromoplexy and involves 2 or more chromosomes. These chromosomes break and then are recombined through non-homologous end joining, forming new chromosomes that can present complex translocations. When we perform a comprehensive analysis of breakpoints and junctions, we can see signatures of these mechanisms that I've just showed you, and we can also see the genomic architecture around these junctions. And this is important because it can help us infer the mechanism of formation, which can then help us understand how cells respond to DNA damage and the nature of the DNA sequences involved in these rearrangements. This analysis also allows us to perform a full characterization of the rearrangements, which then allows us to position the rearranged genomic segments, phase multiple breakpoints, and pinpoint interacted genes or regulatory interactions, which then allows us to correlate the alterations with the phenotype, which is important for follow-up and genetic counseling. The aim of this study is to resolve complex rearrangements using different methodologies, comparing their resolution and potential in detecting structural variants. We start our study with classical cytogenetics analysis. So by performing karyotyping and chromosomal microarray analysis, we were able to select 6 individuals that could present complex rearrangements from the Medical Genetics Center at the São Paulo Hospital. These individuals underwent optical genome mapping, which is a technique that uses ultra-high molecular weight DNA, which is isolated and then is labeled in specific sequences across the genome. Then this DNA is transferred to a chip, which is then inserted into the equipment. And then this equipment linearizes and images these molecules. And these images are then converted into molecules, which are converted then into consensus maps, which are then compared to the reference so that we can detect the structural variants. The optical genome mapping analysis allows us to see an approximate location of the breakpoints. So then after that, we performed long-read sequencing with a lower coverage of 10x throughout the whole genome so that we can precisely locate the breakpoints and characterize the junction points. We used high molecular weight DNA, which is then sequenced until we get hi-fi reads, which present 99.9% accuracy. We used variant colors to do the analysis, but we also performed a visual inspection on IGV. And there we were looking for chimeric reads, which are these breakpoint spanning reads, where these rainbow parts of the read align to a different region of the genome than the one that we are looking at our screen. And here we can see the breakpoint. Having done the entire characterization of the rearrangement, we then perform the karyotype-phenotype correlation, where we associate the chromosome alterations with the phenotype. To do this, we use many different tools as well as information from the literature. So as I mentioned before, we have 6 cases in this study. This is an overview of all of the cases. Here at the top, we have 3 cases that present complex chromosomal rearrangements involving multiple chromosomes, while in the bottom, we can see 3 other cases that present complex genomic rearrangements involving a single chromosome each. Since there is no time to speak about all of them, I would like to highlight case 1, which is a 6-year-old female that presents many different phenotypes, including neuropsychomotor developmental delay, microcephaly, hypotonia, and different facial features. She had her rearrangement first characterized through karyotyping, and this is her result. We saw a possible alteration here on chromosome 12, which could be a deletion or a derivative of a translocation. Then when we performed chromosomal microarray analysis, we saw a 2.4 megabase deletion in the long arm of chromosome 4 and no alteration in chromosome 12. The difference between these results of these 2 techniques is what made us continue the investigation and perform optical genome mapping. This is the optical genome mapping result in the circus plot, where you can see all of the chromosomes. And if we look here into chromosome 4, we can see the call of the deletion. So here at the top, we see the ideogram of chromosome 4. Then we see the CNV call track, the SV call track. Then in green, the reference of chromosome 4, and in blue, the map of our individual. Here at the CNV call, we can see the deletion. And then when we look at the maps, we can see that all of these labels here in this red area are not present in the map of our case. Therefore, we have here a deletion of this region in chromosome 4, just like we saw in chromosomal microarray. If we look back into the circus plot, we see here in the middle these pink lines, which represent translocations, which mean that the patient have a complex chromosomal rearrangement between chromosomes 12, 14, and 21. If we look into one of these pink lines, we see a map representing one of these translocations. So I'll show you one of them that shows the formation of this derivative chromosome 14, where we have here in green the reference of chromosome 14, then the map of our case, and then the map of the reference of chromosome 21. If we look into the blue map, we see that it aligns with the reference of chromosome 14 until a certain point where it starts aligning with the reference of chromosome 21. Here in the middle, where the alignment changes, in this pink-purple area, we have the approximate location of the breakpoint. If we look into all of these maps of these lines, we then are able to resolve this rearrangement. So here we have the 3 chromosomes involved. And for the formation of this rearrangement, we see that we had breaks in chromosomes 14 and 21. And then they joined together to form the derivative of chromosome 14 that I showed you in the previous slide. Then a region of chromosome 12 left its original place, inverted, and joined with chromosome 21, and then with chromosome 14, forming the derivative of chromosome 21. And then both breaks in chromosome 12 joined together to close this gap. So this is the final rearrangement of our patient involving these 3 chromosomes. After resolving the rearrangement with optical genome mapping, we had to go back to karyotype and reposition the derivatives of chromosome 14 and 21 because they were missed in our initial analysis. But when we went back there after the OGM analysis, we were able to reposition and correct the karyotype. After that, we looked into the junction points of the rearrangement. And I'll show you the junction of the derivative of chromosome 14, which is the same chromosome that I showed you the OGM map. So here is a view of the junction, where here on the left we have the region of chromosome 14, and on the right the region of chromosome 21, and we see the chimeric reads. And if we zoom in on the breakpoint, we can see how the genomic architecture is in this area. So here we see in green the region of chromosome 14, and in blue the region of chromosome 21. These two regions don't join together with a simple break and join. Here we see the precise breakpoints. A similar result was also found for both junction points in the derivative of chromosome 21. But for the derivative of chromosome 12, we saw something a bit different. I'll show you here. We have both regions of chromosome 12 shown here at the top, and zooming into junction, we have in brown both regions of chromosome 12 here at the edges of the image, and then in the middle in yellow we have an insertion of a sequence between the two breakpoints. I'll show you more about this insertion in the next slide. Having done the sequencing of the junction, we are able then to infer the mechanism of formation of this rearrangement. So for the junctions in the derivative of chromosome 14 and 21, we saw a simple break and joining, which then leads us to inferring the mechanism of chromoplexy for the formation. This mechanism is the one that I explained previously, where we have multiple chromosomes that break and then are joined together through non-homologs and joining and forming these complex translocations. For the derivative of chromosome 12, we see that we have here a different way of showing the sequence that I showed you in the previous slide, where we have in brown the regions of chromosome 12, and in the middle here we have the insertion in yellow. If we look closely, we see that the insertion is a repetition of the final 12 nucleotides of the upstream breakpoint, which is repeated many times until it finds the other breakpoint. So here we have the insertion of a repeated sequence. This is something we usually see in junctions formed by the backward replication slippage mechanism. In this mechanism, we have DNA replication, and the new strand, which is shown here in black, disengages from the template and slips backward and replicates the same region more than it should. So here in this example, this brown area is replicated twice instead of only once like in the template. Then we can have another backward slippage where we can have the replication of this region again. And then in the final product, we see here this region being repeated many times, just like we see here in the junction of our patient. Having done this entire characterization, we can then look into the phenotype. And going back to the beginning, we saw the deletion in chromosome 4. Interestingly, we were not able to find chimeric reads in the long-read sequencing to show this deletion, but the CNV caller found this deletion. And looking into this region, we see 5 main genes which are deleted, and one of them is the CLCN3 gene, which is associated with neurodevelopmental disorder, with hypotonia and brain abnormalities, which is associated with many different phenotypes, including microcephaly and hypotonia, which are present in our case. And we have also to look to the complex rearrangement to some of the breakpoints. An interesting breakpoint is the one in chromosome 12, where we have the rupture of a gene that is associated with neurocognitive disorders in Mexican individuals. And also other genes in this chromosome band where the breakpoint happened are also associated with the development of cognition. Therefore, this break could be associated with the patient's intellectual disability. Another interesting breakpoint is the one in chromosome 14, where we have the disruption of the regulatory context. So here we can see the enhancer-promoter interactions between the genes and the enhancers. And here we have the PRKD1 gene, which the breakpoint is impairing the relation with an enhancer. This gene is associated with congenital heart defects and ectodermal dysplasia, which is associated with phenotypes including dysmorphic features that are present in our case. Therefore, these 3 genes could be explain at least part of the phenotype. If we see this case, we can see that each technique brought something new to the analysis. So we have some difference between the methodologies. We can highlight that karyotyping in chromosomal microarray analysis missed the involvement of chromosomes 14 and 21. OGM then revealed the full rearrangement, and long-read sequencing precisely located and characterized all breakpoints and junction points. Something interesting to highlight is how far the OGM breakpoints were from the actual breakpoints found by long-read sequencing. And for this case, the average breakpoint difference was 2,641 base pairs. All 6 cases were resolved with optical genome mapping and long-read sequencing. I'd like to highlight very quickly just some of them. Case 4 was the one that presented the least number of breakpoints and junction points, 4 breakpoints and 2 junction points. Despite that, this case presented a complex rearrangement, which is known as duplication, inverted triplication, duplication. And with optical genome mapping, we saw the evidence of a triplication and of inversions at the edges. Then with long-read sequencing, we saw the presence of the duplications, and then we were able to characterize both of the junctions and infer a mechanism of formation as the combination of the fold-back mechanism with non-homologous joining. And we are also able to highlight 2 genes that could be associated with the phenotype. 2 other interesting cases on the other end are case 3 and case 6, which present the most number of breaks and junctions. Case 3 is a rearrangement involving 6 chromosomes that presented 42 breakpoints and 29 junction points. And case 6 is a rearrangement involving a single chromosome that presents 34 breakpoints and 17 junction points. So the number of involved chromosomes does not correlate to the complexity of the rearrangement, as only a single chromosome can present many breaks and junctions. So here again is the overview of all cases which were resolved. And with this study, we can conclude that the complete characterization of the rearrangements with breakpoint mapping and junction point sequencing allowed for the inference of their mechanism of formation, as well as their correlation with the phenotypes. And due to their higher resolution, it would be ideal to perform long-read sequencing with a lower coverage if combined with optical genome mapping to resolve complex rearrangements. I'd like to thank everyone involved in the project and everyone watching. Thank you.